Then loop the histones to make the chromatin fiber. That's still the relatively the stage at which you find them during interphase. And then the loop domains and the heterochromatin here, that's only during mitosis or if the genes are not being used and you want to turn them off. So one way to turn off the genes is condense your chromosomes. At least condense that part of the chromosome. Those genes are not used. All right, so most of the time you're looking at interphase. You look at the cells and it looks like you don't see any chromosomes in the nucleus because they're not condensed. So what is happening? So during interphase, there's gap 1, gap 2, and the S phase in between. S stands for synthesis. That's when DNA is replicated. Depending on how much DNA you have, S could be long or it could be relatively short. So different species have different length S's. G1 and G2 used to be called, well, they're still called gaps because we didn't really know what was happening, but it turns out there's a lot of biochemistry that is happening in both of these. So you're getting ready for DNA replication during G1, you're getting ready for mitosis during G2. So lots of biochemistry happening in those two phases. It's just not visible under the microscope. The other thing that you should note is this G0, G0. Some cells can leave G1 and enter this arrested phase. So they will not go through the rest of the cycle. They just won't divide. And they can be stimulated to go back into the cycle and then go through division. And the regulation of what causes them to leave the cycle or re-enter the cycle is very interesting, especially if you're interested in cancer biology. And your textbook does have some information about that, and we are not going to talk about that. So if you're interested, read it on your own. We do not. You will do some of that in Bio 229, but we're not doing it here. So, interphase, G1. DNA is packed on histones, but not much more than that. You just get your regular chromatin fiber, because that's where you get ready for DNA replication. And you can't have a condensed chromosome if you're going to replicate the DNA. So the DNA can't be condensed in that phase. During the S phase, the synthesis phase, you replicate your DNA. After that, you're getting ready for mitosis, so the chromatin coils and coils, chromosomes begin to condense, and all the other machinery that you need for mitosis begin to form as well. So artificially, we divide mitosis into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. And when you take upper level courses, you see that each of these is then subdivided into other phases. We don't do that here. We would just look at it very superficially, just looking at these 5 phases. So we don't, if you know about the leptotene and glycogen and all of those, we don't do that in this class. You'll do that in upper level courses. Now, one thing to remember is that this is all artificial by us. To try to categorize what happens in each step, you have to remember that this is continuous. It's not like you have prophase, then you stop. Then you go into prometaphase, then you stop. This is all continuous. And things might be happening simultaneously, so telophase and cytokinesis overlap all the time. In some cases, you don't get cytokinesis. You go through mitosis, and you go through mitosis again, and you go through mitosis again, and then you have multiple cytokinesis happening at the same time. So it's not like you have to go through this on cytokinesis before you can do this again. But for simplistic, to make it simplistic for you, we're going to pretend like everything happened linearly that way. So I'm not going to use the figure from your textbook. You can look at that in your textbook. We're going to look at a different figure. If I could figure out how to turn off the lights, it would look a lot better, but I can't, so. Turn off lights, which is that. Where? Go show you. So much for the light switch. All right, so what do you have? Yeah. No, I need to turn off these lights, not those lights.
Oh.
All right, so what you have here are nicely stained DNA as well as microtubules, so you can see the formation of the spindle fiber and the condensation of the chromosomes. So during prophase, what you see is the condensation of the chromosomes. So before prophase, the chromosomes are not visible here. You can see the nucleoli in the nucleus, but no individual chromosomes are visible. So what happens is the condensation of the chromosomes allows you to see these squiggly lines. Those are the chromosomes. And then the chromosomes condense and condense and condense till you get the two sister chromatids attached at the centromere, nicely visible like an X-shaped chromosome. At the same time, you're getting the formation of the spindle fibers. The spindle fibers are made up of microtubules and other proteins. And in animals, you have centrosomes, which are shown here. These centrosomes replicate before prophase, and then they migrate to the opposite poles of the cell, and then the spindle fibers actually radiate from them, from the aster here, to eventually attach to the proteins on the side of the centromeres called the kinetochore. And we're going to talk about that briefly as well. But you don't have centrosomes in plants or fungi. So you have them in animals, but not plants or fungi. So not all eukaryotes require centrosomes for this to work. And it turns out that you can remove the centrioles here, and it still works in animals. So it's not certain that this is actually essential for animals either, but they have them. Right. So the microtubules have to attach to the chromosomes so that they can align them in the cell. So by the end of prophase, the microtubules have attached to the chromosomes, and you have protein complexes on either side of the centromere called the kinetochore. And did I put that on here? Yes, I did. Kinetochores. So these are proteins on either side of the centromere to which the microtubules can attach. And here's a diagram showing you the condensed chromosome, kinetochores on either side of the centromere, microtubules attached to the kinetochores. Now there are groups of proteins that keep the two centromeres together, and it used to be thought that they were cohesins, and it turns out cohesins are part of the protein complex that keep the chromosomes together. So there are cohesins and other proteins, so they're a little more complicated than we used to think. And so the proteins that hold the centromeres together have to be cut at some point so that the two sister chromatids can separate from each other. So let's go back to the diagram here. At this point, sister chromatids are still joined. Microtubules attach to the kinetochores. And during prometaphase, the microtubules start aligning the chromosomes, so they start pulling and tugging and pulling the chromosomes, so that by metaphase, the chromosomes are aligned in the middle of the cell, in what we call the metaphase plate. Now, when the chromosomes have aligned, then the cohesins and other proteins that hold the sister chromatids together will be cut, and that's the beginning of anaphase, during which the sister chromatids will be pulled to opposite poles of the cell. So you had 46 chromosomes during metaphase and during prophase, and each of those chromosomes had two sister chromatids attached to the centromere, because each of those chromosomes was a replicated chromosome. During anaphase, you separate the sister chromatids. At that point, each of them becomes a separate chromosome. We no longer call these sister chromatids. They are now independent chromosomes. So you start with 46 replicated chromosomes. Now you have 92 normal chromosomes. So far, so good? All right, so during anaphase, the chromosomes, the new chromosomes, which used to be sister chromatids, joined at the centromere. They're no longer sister chromatids because they've been separated. They move to the opposite poles of the cell. And during telophase, what happens is that this nuclear envelope reforms, the spindle fiber disintegrates again, and the chromosomes begin to decondense, and telophase usually overlaps with cytokinesis, which means the cell division begins usually while telophase is happening. So you can see that with the fluorescent slides above, so you can see the formation of the spindle fibers and condensation of the chromosomes. Here you have the microtubules attached to the chromosomes, but they haven't aligned yet. Now the chromosomes are aligned at the metaphase plate here, with the microtubules going to the aster ends in both directions. Here the sister chromatids have separated, and when anaphase has started, and now the two new nuclei are beginning to form. Question in the back.
What is the last phase called?
During telophase? Yeah, telophase and cytokinesis can overlap. So while telophase is happening, the new nuclei are forming, the cytoplasm begins to divide. All right, so this should be basically a review from high school bio, for those of you who took high school bio. So the mitotic spindle, remember, it's not just microtubules, there are other proteins involved. And the centrosomes are important for animals. They're not important in plants or fungi or algae. Centrosomes replicate during interphase. You get two copies, they move to the opposite poles. Spindle fibers form from the centrosomes. They're called the mitotic center or mitotic organizing center. There are two types of